Hi, I'm Elvisha Dhamala, and I'm here to talk about sex-specific brain behavior relationships in healthy and clinical populations. So I want to start by, first of all, thanking you all for joining us today for this wonderful symposium. I want to thank all of the organizers for giving us this opportunity to present this work, and I want to thank all of the other speakers that have presented some really exciting work so far. So with that, let's get started. White matter fibers in the brain directly and indirectly connect different brain regions, and these structural connections facilitate the flow of functional neural activation signals between brain regions. Together, these structural and functional properties of the brain underlie our unique and individual behaviors. But even with this idea of some aspect of brain leads to some aspect of behavior through some mechanism or intermediate relationship, we have to recognize that there are several different reasons why a sex difference may or may not exist. Specifically, you can have differences in the brain, the behavior, or some relationship between the two. And in my case, I'm primarily interested in leveraging machine learning models to understand how brain behavior and brain behavior relationships are shared or unique across the sexes. So let's break that down a little bit, and then I'll present some brief results on some of the work that I've done in this area. Speaking of sex differences in the brain, throughout the lifespan, it's been well established that males have greater volumes than females, about 10% or so, in terms of gray matter, white matter, subcortical gray matter, and ventricular volume. These differences emerge at different points throughout the lifespan, but tend to persist. But the differences are not just in terms of structure. There are also notable differences in brain function. This study using the Philadelphia neurodevelopmental cohort showed that females are more likely to exhibit stronger within module or within network connections, whereas males are more likely to exhibit stronger between module or between network connections. More recent work has shown that functional topography also differs between males and females, and it can actually be used to correctly classify males and females with an accuracy of almost 83% and an AUC of 0.94. And the most important networks for these classifications are different heteromodal association networks, such as the ventral attention network, the default mode, the frontoparietal, and the dorsal attention. And what's even more interesting is that those same networks that distinguish males and females are also implicated in a range of different behaviors. So this study from Thomas Yo's group demonstrated that functional connectivity can predict a range of behaviors linked to cognition, personality, and mental health. And the connections that are most important for those predictions are found in different heteromodal association networks. So this tells us that there are sex differences in the brain, and the areas where those differences exist are also associated with our behaviors. So with that being said, I'm curious in understanding if there are potential differences in the underlying brain behavior relationship. Well, we can leverage machine learning and predictive modeling approaches to study that. Predictive modeling approaches can be used to study relationships between brain and behavior at a group level, and then make predictions about behavior at an individual level. And there are different types of analyses that we can perform using them, and today I'm going to focus specifically on predictive analyses. Predictive analyses can be used to identify relationships between neurobiology and behavior, which I'm using here to mean things like diagnostic categories, dimensional symptom profiles, cognitive abilities, and personality traits. And once we establish those relationships between brain and behavior at a group level, we can then actually predict an individual's specific behavior based on their specific neurobiology. But when developing these predictive models, there are many different methodological choices that we must make that can directly and indirectly affect the model's performance and interpretations. Some of these choices include the neuroimaging modality and state that we choose to base our predictions on, the parcellations that we use, how we transform our data, how many subjects we include, whether we use population-specific or general models, the algorithms that we use, and finally, the phenotypes that we're seeking to predict. And today, I'm specifically going to talk about how we can leverage population-specific modeling approaches to understand sex differences in brain behavior relationships. So basically, when we're developing these machine learning models, we can choose to base them on the entire sample or the entire population that we have. Or we can split the sample into different groups based on some category or some criteria like sex. Then we can actually model the relationships in each group or sex separately and then compare how similar or different they are. And not only that, but we can also see if models that are trained in one sex will generalize to another sex and vice versa. So it's a pretty straightforward and simple approach, but it's a great way to understand and quantify multivariate sex differences in brain behavior relationships. So now I'm going to briefly talk about a few different studies that we've done implementing these approaches to study sex differences in brain behavior relationships, and then I'll finish off with some general conclusions. So in this first study, we evaluated relationships between functional connectivity and cognition, and we wanted to look at whether the same or different functional connections predict cognitive abilities in males and females. So specifically, we used data from the Human Connectome project, and we trained models based on functional connectivity in either males or females, and then evaluated whether they could accurately predict different aspects of crystallized and fluid cognition. These are our prediction accuracy results, and the different scores are shown for models trained and tested in males, shown in purple, models trained in males and tested in females in blue, trained in females and tested in males in green, and trained and tested in females in orange. And the colored in violin plots denote that they perform better than chance. And we see that functional connectivity comparably predicts crystallized cognition, as well as picture vocabulary and reading, comparably in males and females, regardless of who the models were trained on. But functional connectivity only successfully predicts fluid cognition in males, if at all. So this suggests that if there might be underlying sex differences in the strengths of these brain behavior relationships in terms of functional connectivity and cognition. So next, we looked at those specific brain behavior relationships that the models were capturing and summarized them into positive and negative associations between connectivity and crystallized cognition on the top and fluid cognition on the bottom. And the relationships are shown for females on the left and males on the right. And overall, we found that the same functional connections between different cortical-cortical connections are associated with cognition in both males and females. And even though certain predictions were only significant in males, it suggests that the underlying brain behavior relationships are still the same when it comes to functional connectivity and cognition. But without running these analyses, we wouldn't have been able to know that for sure. And this also doesn't mean that this result of the same relationship for males and females holds for all brain behavior relationships. So in this next study, we actually influenced how ICV correction, or intracranial volume correction, can influence how we interpret regional brain behavior relationships, and particularly when we're thinking about these relationships across the sexes. So first, we started off by looking at whether ICV itself could predict cognition, and found that it can predict crystallized cognition in both males and females, but could only predict fluid cognition in females. In this graph specifically, we have the prediction accuracies for predictions in HCP, or the Human Connectome Project, at the top, and for ABCD, or the Adolescent Brain Cognitive Development Dataset, on the bottom. And we ran all these analyses in both sexes, shown on the left, just males in the middle, and just females on the right. And the asterisks here denote that the predictions were better than chance. And we see that in both HCP and ABCD, we can accurately predict more cognitive abilities, especially within the fluid domain in females compared to males. So this is actually the opposite of what we observed previously with functional connectivity and cognition, but here it seems that there's actually a stronger relationship in females than there are in males. Next, we looked at whether proportional ICV correction affected how accurately structural properties of the brain could predict cognition. Specifically, we trained models to predict cognition based on surface area, shown on the left, gray matter volume, shown in the middle, and cortical thickness, shown on the right. And once again, we have results from HCP on top and, HCP males on top and HCP females on the bottom. And the violin plots in green are based on uncorrected measures, so uncorrected measures of surface area, gray matter volume, and cortical thickness, whereas the ones in orange are based on corrected measures. And we can see that the ICV correction seems to have a greater influence on the prediction models in males than in females, as denoted by the difference in performance between the split violins and the significant ones denoted with the black asterisk. We also evaluated the same thing in ABCD data and found similar and actually even stronger results. Here, we see that ICV correction has a greater influence on the predictions in males than in females, and especially for the predictions based on surface area. So this now tells us that when considering a confounding factor like ICV, even when it's within a single sex, there might be differences in how that is influencing the underlying brain-behavior relationship. So together, these results suggest that when considering different variables that might confound a relationship, that confounding component may actually be different across the sexes. So it's worth investigating and identifying if that's the case, so you can get more accurate and reliable results overall. And finally, most recently, we've done some additional analyses looking at how functional connectivity can predict different behaviors linked to psychiatric illnesses. And specifically, we were looking at whether functional connectivity could predict a range of behaviors that are associated with externalizing and internalizing symptoms of psychiatric illnesses, and how those were similar and different across the sexes. Specifically, we showed that the same functional connections and heteromodal association networks that predict internalizing behaviors in females actually predict externalizing behaviors in males. And if you're interested in this work specifically, I'm not going to talk any more about it, but I encourage you to come check out my poster on it. So with that, I'm going to briefly summarize the main conclusions that I hope you get from it. And with that, I'm happy to take any questions that you might have by email. So overall, based on this talk, I hope that I've been able to show you that these population-specific predictive modeling approaches can be used to evaluate similarities and differences in brain behavior relationships across the sexes. These results are complementary to those obtained with general models, by which I mean models that include the entire population, that can capture brain behavior relationships that are largely shared across the sexes. Whereas these population-specific or sex-specific models can be used to understand sex-specific brain behavior relationships that might otherwise go undetected. And even when we're considering brain behavior relationships that are the same across the sexes, it's important to recognize that they may be differentially influenced by other factors such as ICV or anything else that might be considered a confound. And finally, it's important to conduct these analyses and understand these relationships and what might be influencing them, because these insights can actually guide the development of sex-specific biomarkers for diagnostic tools and therapeutic interventions. So based on these insights overall, I encourage you all to perform sex-specific analyses in your future predictive modeling work to evaluate if a given relationship is the same or different across the sexes. And with that, I'd like to thank you all for this opportunity to present this work. And I also want to thank my lab and my funding sources, as well as the different researchers and the labs that have contributed to these studies. And I also want to mention that if you're interested in these sorts of questions and understanding sex-specific brain behavior relationships, I am hiring postdocs to join my lab that I recently started. So please do reach out, and I'd love to chat more, and you can also find more details through that QR code. Thank you.